Hello, everyone, and good afternoon. My name is Carl, and together with me is Gail. And today we're going to be talking about the ICH as well as information about generic drug development and approval. So, the world is evolving faster than ever, and there are new diseases and disorders, and new drug discoveries are equally getting in higher demand. And due to this rise of new drug discoveries, there's always a huge demand for harmonizing the medicinal products. So, in order to control and maintain the standards for quality, safety, and efficacy of each medicinal product, let's see today the organization that takes the responsibility to harmonize the medicinal products, which is the ICH. ICH stands for International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. And for us to get a better understanding of ICH, let's answer these five questions about it. So, what is ICH? ICH is an international non-profit organization which aims to deliver harmonized guidelines for global pharmaceutical development. When was it found? It was found on April 1990. And why was ICH founded? This is to harmonize the technical requirements in and to ensure quality, safety, and quality of medicines, and to minimize the use of animal testing and prevent the duplication of clinical trials and others. The ICH birthplace is in Brussels, Belgium, and the current ICH secretariat is located in Geneva, Switzerland. So, who are the founding members of the ICH? The founding members of ICH are the Europe, United States, and Japan. So basically, ICH brought together government regulators and drug industry representatives from the US, Europe, and Japan to make the international drug regulatory processes more efficient and uniform. It should be noted that the regulatory systems in these regions share the same fundamental concerns for safety, efficacy, and quality of drug products, the goals of which are to make new drugs available with minimum delay, to minimize unnecessary duplicate testing during the research and development, and to develop guidance documents that create consistency in the requirements. And now let's talk about the ICH process for guideline development, which involves five steps. Step one is to prepare the consensus draft of the technical document based on the objectives set out in the concept paper, as the second step involves the ICH parties' consensus of technical document, which is documented and is adopted as a draft guideline by the regulatory members. Step three occurs as a three stages. First as the consultation, second as the discussion, and then the third is the finalization. The next step, step 4, involves the adoption of an ICH harmonized guideline, and lastly, step 5 involves the process of implementation. And now for the major subjects of the ICH guidelines. These guidelines are classified into four categories, which are the quality guidelines, the safety guidelines, efficacy guidelines, and the multidisciplinary guidelines. The quality guidelines is further classified into 14 separate guidelines, and it discusses about the pharmaceutical quality. It deals with stability testings on new drugs, analytical validation, impurity studies, pharmaceutical development, life cycle management, quality risk management, and many more. Safety guidelines, on the other hand, is further classified into 12 separate guidelines. The purpose of these guidelines is to uncover the potential risks like carcinogenicity. So here we can also find studies like genotoxicity studies, pharmacology studies, immunological studies, and even non-clinical pediatric safety, which is proposed to provide direction on the non-clinical safety important to support a pediatric development program. Next, we have the efficacy guidelines, which are classified from E1 to E20, and they discuss about the design and conduct safety reporting of clinical trials. It involves pharmacovigilance, which guides the key aspects of clinical safety reporting. It also involves dose response studies, statistical principles, and even ethnic factors, which addresses the intrinsic characteristics of the drug recipient and the extrinsic characteristics associated with environment and culture that could affect the result of clinical studies carried out in regions. And finally, we have the multidisciplinary guidelines, and they talk about the topics that are unique and that doesn't cover from the quality, safety, and efficacy categories. The MD guidelines include drug interaction studies, gene therapy, non-clinical safety studies, and bioanalytical methods of validation, which apply to the validation of bioanalytical methods and study sample analysis in non-clinical and clinical studies. So based on what we have learned so far, we can definitely say that ICH possesses a lot of value and is beneficial. It helps in reducing the development times and resources. It also helps for easier new drug launches. ICH helps to have a recognized global standard. In short, ICH helps in bringing life-saving treatments to patients faster, and it helps the pharma companies to develop standard medicinal products.
Generic drugs are copying that one company makes of a brand name that was developed by another company. Generally, generic drugs sell at lower prices, and it is in public's interest to get generic drugs to the market quickly. But, unlike any other scientific and regulatory process, approval of generic drug takes time. It takes the FDA time to review the complex information needed to make or demonstrate that a given generic drug can be substituted for a brand name drug that is that it copies. And that time also it depends on the complexity of the drug product and the completeness of the application. In the characterization stage, some generic products are challenging to develop because of the complexity of the reference product. Complex reference product may include drug substances with many potential active molecules. Molecular diversity can arise in products with a natural source, origin, or even in synthetic molecules that are polydisperse mixtures. Other challenging reference products include products with complex supramolecular structure, such as iron complexes, and products containing liposomes. The use of nanotechnology in drug delivery will lead to even more complex pharmaceutical structures to characterize, and the sponsor may need characterization than the than the originator to make a copy of these products. In the formulation development stage, generic formulation may be developed that do not pass the bioequivalence study or met the other requirements of formulation development must be repeated. These failures can sometimes be linked to the inability of the solution to evaluate proposed formulation and process during development. For new or complex dosage form, the formulation and product development stage can fail because of an inability to identify the cri- critical quality attributes of new or complex dosage form. The type of bioequivalence study required can determine whether the development process is economically and scientifically feasible, expensive, intensive, or unpredictable. BE tests can limit the development of generic product. A higher variable drug may require a large number of subjects using the usual FDA recommended study. However, the biggest limitation to generic competition is when FDA requires a clinical endpoint bioequivalence study. Many generic companies do not develop products that need clinical endpoint bioequivalence study because of the relative cost, time, and risk of failure involved. Finally, a generic drug that demonstrate bioequivalence to the reference product can be delayed in reaching the market because of the final scale-up step in the generic drug development process. Problems with scale-up include waste, commercial batches, failure to met specification, and process variability. These problems may require the sponsor to reformulate the product or revise the development. Here is the under requirements for approval of generic drug products or must. First, contain the same active ingredient as the innovator drug. Thus, an active ingredient may vary. Second one, the identical in strength, dosage form, and the route of administration to the innovator drug. Third, be a bioequivalent drug. Fourth, meet the same standard for identity, strength, purity, and quality. Lastly, it must be manufactured under the GMP.